Welcome to the next episode of CPN TV. I'm Alyssa Betts, an associate professor and extension plant pathologist with the University of Delaware. And today I'm joined by Alison Robertson, who's a professor and extension field crops pathologist with Iowa State, and Erin Sparks, an associate professor at University of Missouri, specializing in plant developmental biomechanics. And today we're going to be talking on this theme of robotics to evaluate management practices in corn. And Alice and I are both pathologists, and Erin brings a little bit different perspective. But we'll get things started thinking about this idea of root rots, seedling blights, and stalk rots. And these are issues across all of the U.S. Every couple of years, we put together kind of summaries of some of our estimated disease losses. And within these groups, over the 2020 to 2023 period, we saw about 639 million bushels of loss that came from stalk rots, over 82 million bushels from our root and seedling blights. So collectively, these make quite an issue for us pathologists and farmers to deal with. When we think about how we kind of look at and evaluate some of these in the field, a lot of times we're really left with, just seeing things after they've already happened. So here, on the left side, you can see a young plant that was showing some early disease. We're a little too late. That plant is probably not going to make it anymore. And, of course, if we have wind events or other stalk rot issues that lead to something as severe as this picture, we're a little late in that field as well. We do have some tools in situations where maybe not quite so extreme as this, where we can walk through and do tests that we call either pinch tests or push tests. And there we're kind of just manually trying to evaluate, what does that stalk integrity look like? Are we seeing signs or symptoms of disease? But this brings in some of the tools and arsenal that Erin can share with us of maybe what are some things us pathologists can do that's a little bit outside the box.
Yeah, thank you, Alyssa. So my lab is really interested in plant mechanics. We work primarily in lodging, and so we've developed a lot of tools to evaluate plant mechanics in the context of lodging resistance. But what you're going to hear today is how we're using those tools in different ways and different ways to evaluate plants. And so my lab has and continues to develop a suite of different tools that we'll talk about today. Primarily, you're going to hear us talk about what we call the pusher, which is a device that evaluates stalk mechanics in an automated way. And then another device we call the Smurf, which stands for sorghum and maize under rotational force. And this is a device that's measuring the anchorage of the root system. Again, we're working on a holistic assessment, so ways that we can evaluate all parts of the plant. And so a newer device we call the spider, which is looking at kind of how the soil moves and integrating that soil piece. So right now, and what you're going to hear about today has really been about the plant, but we understand a plant doesn't grow in the absence of soil. And so we're also working to expand this to look at soil mechanics as well. When we talk about mechanics, what we're really talking about in all contexts is applying a displacement and measuring a force. And so that displacement can be applied in different ways. You could do a tension test or a bending test or a compression test, and then you're measuring the force. And what happens in mechanics is you first have a range where the plant will act like a rubber band. It's called the elastic range. And this is the range that we mostly work in because these are non-destructive. So you can bend a plant, and if you release it, it will go back to its original conformation. Now, if you were to continue to displace that plant, you'll end up with what we call plastic deformation or permanent deformation. And this is where we start to see the breaking or cracking of the plant material. And so for the purposes of this talk, we're talking about elastic deformation, these very small movements, which you'll see in a couple slides, where the plant is able to completely recover and we're not damaging it. And so here is the pusher I mentioned. You're going to see a video here. And so this is, there's a load cell which measures forces, and it is advancing slowly forward and it's moving the stalk. And as it moves the stalk, it's measuring those forces. So this is, again, the concept of applying a displacement and measuring force. And so here we'll get basically the graph I showed you on the previous slide, where you can see the stiffness of the plant. And now the next thing we're going to show you is the Smurf. This attaches to the base of the plant and it keeps the stalk rigid so that you're isolating the root system. It again is applying a displacement by levering over that stalk, and it's measuring the resistance of the root ball to rotation. And so both of these are measuring slightly different things. As you'll see in this talk, they're often really highly related.
Awesome. So Erin and I have worked together for a number of years in trying to see, can we use some of these tools in different diseases? And in particular, one we were interested in was pythium root rot in corn. And when we think about pythium in corn, there's death that happens early on, but we can also get these plants where they're stunted, but they live. They can make it through the season, but they're really not doing that well over the course of the season. And so over the past couple of years, we've been trying to learn more about these pythium species, and they're not just a problem for us in the Mid-Atlantic. And we'll let Alison kind of talk about a little bit of the history of pythium in Iowa as well.
Thanks, Alyssa. So yes, we've had pythium in Iowa, probably started reporting to be a problem kind of around about 2010 when we had some damping off. And the data that I'm showing here is from a survey that we did in 2012, which was where we went out and we collected corn plants from commercial fields. And then what we did was we recovered the causal organism from those plants. And so what you'll see here is that we had 9 species of pythium that were recovered from those plants. And by far the most prevalent species was Pythium tolulosum. But we also had several others, Sylvaticum, Heterothalicum, Irregulari will probably sound familiar to some folks out there.
And so this study, really kind of set the stage along with some other work that was happening in the Midwest of there's a lot of diversity out there in pythium. So when I came to Delaware and we started seeing problems in corn, one of the first things I was really interested in is what do we look like in the Mid-Atlantic as far as species recovered? And we saw that, again, there's a lot of species involved. We've found over 14 so far. But what's really interesting is our makeup is quite different. And so for us, Pythium cramonicola is the dominant species we've been seeing, which, wasn't really even showing up in your surveys, Alison. And, if we look at some of our other colleagues through Ohio and other areas, we see some gramonicola out there, but that does seem to be a little bit more prominent here in the East and has been one that my lab's been focused on. But at the end of the day, all of these pythiums, no matter what species you're looking at, kind of have similar results as far as what we see in the corn plant. And one of the things we were interested was like, we know we have plants that die, but when these plants live, what does that plant do through the season? And do we ever have any ways that we can try to maybe change the course of that plant? But first, to do that, we really wanted to track some of these differences. So we were marking plants when they were about V3, and we would have a symptomatic plant and a partner next to it that was kind of a healthy looking or non-symptomatic plant. And we tracked these plants over the course of the season. And at the end of the year, we'd come through, dig out these root systems, do kernel counts. And you can see from the pictures in this, it's pretty dramatic. Our plants that had pythium symptoms at the beginning of the season versus not. And a lot of this has to do with the shading and the other just range of challenges that these stunted plants have to face throughout the rest of the season. So, from our end, we were culturing, kind of understanding these pythium species, looking at some of these factors such as the shoot characteristics, the root characteristics. But we pulled Erin in on this, and her team was also able to look at things from some of the biomechanic perspectives.
Yeah, and so to Alyssa's point, we wanted to say, could we quantify these end of season differences with the mechanics of the plant? And so throughout the rest of the presentation, the graphics you'll see, the pusher icon is there to indicate that that's looking at the stalk versus the Smurf icon, the root system. And so across multiple years, 22 to 24, in the stalk, we repeatedly see at the end of season that the Pythium plant or the Pythian early infections basically grew up to be weaker for both the stalks and the roots. But to Alyssa's point at the very beginning, it's way too late for us to do anything at this point. And so we started to ask, okay, can we go earlier and earlier? Are we able to start to develop predictions of how these will be at the end of season? And so we went out and we were kind of measuring these weekly. And one of the interesting things that came out for us is I'm going to walk you through this graph a little slowly. So this is looking at root system stiffness, so that root resistance to bending. And so on the left, what you have is one of our really early season measurements. And you can basically see in blue that the Pythium plants are all uniformly weak. And whereas the control plants, we saw variation in how much root establishment. So you can think of this also, even though it's mechanics, it's really related to how well the root system is established early season. And what we saw then is a week later, so in these early time points, there was no relationship to yield, right? All the Pythium plants were weak, regardless of the end of season, how well their Alyssa talked about the kind of what did you talk about? Kernel number.
Yeah, kernel
Thank
counts
you.
and yield estimates.
Kernel
Yep.
counts. And so we were looking at these estimated yields relative to these early season. But what was interesting is a couple weeks later, we now saw this really nice relationship with the root system establishment of Pythium plants. And so basically what this suggests to us is the root systems that can recover are more likely to have higher yield. So if your root system stays weak, you end up with very low yield. If your root system is able to recover, and at the some of these Pythium plants are actually recovering quite well, and that they're in the range of some of our lower control plants. And so this is something that we've recently started to talk about of, is there something we can do to promote root recovery that will allow us to gain yield from these early season deficits? Now, this is completely different in the shoot. And so now in the stalk mechanics, we see a relationship regardless. So these, again, the left panel is this really early season evaluation, and the right panel is a couple weeks later evaluation. And it seems that the stalk mechanics is always related to end of season yield. So if you have a stronger stalk, you're able to recover yield a little bit better. And that didn't seem to matter when we made those measurements. And so even though I said a lot of the mechanics is related, looking at these in the context of disease and recovery from disease, we're able to start to understand some of the nuances in how a plant can recover, with the idea that we could test potential interventions of ways to help these plants recover.
And part of why we're so invested on, can we help these plants recover is if we look at some of the yield impacts from those yield kind of kernel counts and estimates we've done, say, for example, we look at $5 corn. If 5% of that field has Pythium symptomatic plants, so these stunting and they're just growing up to be a bit runtier, we're looking at almost $35 an acre that's lost. And this is not counting the Pythium plants that have, went on to kill those plants early. So there's still losses in addition to this that can be from stand loss, but these are just those runty plants. And as Erin mentioned, this is kind of ongoing. We're trying to target these things earlier and play around with, are there some intervention, maybe some precision ag type of techniques that we can use. And so we're not ready to talk about those results today, but this is part of a 5-year AFRI project that Erin and I are working on. And so hopefully by the end of the project, we'll continue to have a few more updates there. And, Alison mentioned she started seeing Pythium around 2010. For us, 2018 to 2021 was like really wet springs, a lot of Pythium. And so one of the things we've been wondering is why is it we're maybe seeing more pythium? And so it can be from a lot of factors. We have a lot more no-till agriculture. We have had some really wet springs that do favor the pathogen. But one of the other things that we've been seeing too is an increase in cover crops. And so with cover crops, these are often, being planted in the fall, maintained through the winter. We're not growing these for a harvestable purpose, but there can be a wide range of benefits for soil health, for weed control, for runoff. Um, so there's a lot of benefits and reasons why folks are interested in growing cover crops, but there can be some challenges as well. We do have some costs associated with establishing them. And especially with kind of from the disease side, we can sometimes see these green bridges, or we're leaving roots that are in the ground that these pathogens can kind of move from this rye or overwintering crop into our crop of interest in the case of corn. And then we do have other things, slugs, other issues out there that we won't be covering today. But one of the projects that kind of happened in recent years that was incredibly comprehensive was a SAS CAP project. And Alison, I'll let you introduce that since you were kind of one of the main leads on this project.
Thanks, Alyssa. Yes, so the Precision Sustainable Agriculture Project was done over a period of 5 years. It was a very extensive project that developed a lot of tools for cover crops and monitoring different things, but a very, very small part of this project was what we called Common Experiment 2. And in this project, we looked at the effect of cover crop termination timing on seedling disease and then insects, pests, and beneficials, and also weeds. The exciting thing about this research is that it was done in 16 states across the US, and you can see the states that were where the research was done listed there. The two guys on the left, Steve Mirsky and Chris Rehberg-Horton, were the PIs from North Carolina and USDA. And then I led the seedling disease part of the project. John Tucker at Pennsylvania led the insect side of the study, and then Colleen Chase down at Florida led the weed science part of the study. And so the one thing that we did in this study was we tried to keep everything as similar and as possible across the states. And so that meant everyone was on a corn soybean rotation. Everyone used no-till. Everyone planted cereal rye as their cover crop. Everyone terminated that cover crop with glyphosate. We all put down nitrogen at planting, although the amount of nitrogen varied a little bit among the states, probably from 50 to 70 pounds at planting. So everything was very, very consistent. And we were trying to figure out, is termination timing, or the biggest question we had was with regards planting green, because in Iowa, we had shown that planting green led to increased risk of seedling disease and therefore increased risk of yield drag. And so we wanted to be able to see, is this a problem in Iowa or is it a problem with the system? So the treatments that we had an early termination timing, which we nicknamed planting brown, and that was where the cereal rye was sprayed with glyphosate 2 to 4 weeks before we planted the corn. And then we had an at planting termination, which we referred to as planting green, and that's where the corn was planted into green cereal rye that was then terminated then, or on that day of planting, or within a day or two of planting. And then we had the late termination, which we called planting green brown, which was where that cereal rye crop was terminated or sprayed with that herbicide 3 to 7 days before planting the corn. And we knew from research in Iowa that definitely increases your risk of seedling disease. So it was kind of like our positive control. We also had a negative control in there where we had no cover crop present. So if we look at the results, they're a little bit complicated. As as you can see from that chart on the right. But over 3 years, we have the different states and the amount of root rot severity in each of those states. And so what you can see is that the severity of root rot varied across the years and also across the locations. So what I'm particularly interested in is the planting green, which was where the cover crop was terminated around about planting, and the planting brown, which was where the cover crop was terminated at least 2 weeks before planting that corn. And what we can see is that at 12 of the 15 locations, we had more seedling disease when we planted green compared to when we planted brown. In 2021, we had more seedling disease in planting green in 2022 at all the locations, and then at 9 of the 14 locations in 2023. So this is exactly what we'd seen in our research in Iowa. And so a lot of farmers these days are now planting green because it's easier, right? You just go into the field when conditions are correct, and you can get that planting done. And if you have to terminate that cover crop 10 days plus ahead of planting corn, you never know what that weather's going to do. The other thing, of course, which is probably more important than seedling disease to farmers, would be the corn yield, right? And so when we compared the corn yield of the planting green to the planting brown, once again, we saw it varied across the locations and across the years. But if we summarize that, the relative yield of the planting green was less than that of planting brown at 10 of the 13 locations in 21, 11 of the 14 locations in 22, and 10 of the 12 locations in 23. So planting green not only increased seedling disease, but also increased the chance that we would have a yield drag.
So that was a really great project that had so much comprehensive work that came from it. And I'll share just kind of to wrap up one project that also was on the theme of cover crops and rye shading, but this one was a little bit different. So kind of knowing, what this SASCAP project was kind of building, and with the introduction of some of the new short stature corns, we had some questions regarding, what can be the role of rye shading on corn roots and shoots in these traditional and short stature cornfields. And this was an AFRI-CARE project that, again, Erin and I were working along with one of our other collaborators, Dr. Jared Miller, who's an agronomist. And this one, one of our big questions is we'll sometimes see this response in corn that's grown in these cover crops where it grows quickly. And what was really interesting is this short stature corn, it kind of, it's, not to personify it, but it's almost like it knew it was going to grow up and be short. So it really jumps out of the ground quickly, and particularly if there is a cover crop involved. So this is kind of just looking at some of those shoot heights across the bottom. You can see when there was no cover, an early termination and a late termination. And in this first A panel, we're looking at the short stature corn, and in the B panel, the traditional stature corn. So, we see that the short stature corn actually, again, would get a little bit taller than expected very early on. And that late cover crop termination would result in kind of a lankier, taller corn seedling. But interestingly, by the end of the season, these differences kind of went away and everything sort of evened out in the end. And again, we were really interested in bringing in Erin's expertise of, how can we use some of the biomechanics to understand if these cover crops are impacting any of the corn stature and maybe some of the stalk or root characteristics.
Yeah, and to Alyssa's point, that early kind of growth is characteristic of a shade avoidance response. And so when we started this project, we thought that plants would end up being weaker overall because we know the more densely you plant, the more you have a shade avoidance response, the increased likelihood of lodging, and basically weaker stalks. And so we thought that perhaps this this early growth would persist and would cause weaker plants. To Alyssa's point, it seems that it did not persist. And by the end of season, plants had completely equaled out. And we saw that again with all of the mechanics. And so we again were measuring weekly or bi-weekly, depending on the weather, the mechanics of these corn plants across multiple years. We were measuring shoot and root, and it turns out that it's all pretty boring, which is a great news, and that we weren't really seeing any differences in the end of season, early season mechanics of these plants based on cover crop treatment. That being said, there are some differences between short and tall corn. The lines that we had weren't directly comparable, and so we weren't, we're not going to make too many broad conclusions here, but short corn was released with the idea that it's going to be stronger and it's going to withstand some of the storms like the derecho that happened in Iowa. And so we were intrigued that we saw basically no response in either of these. And yeah, it's always great when your initial hypothesis is wrong and it turns out to the benefit.
Yeah, I mean, I think, Erin, this is really exciting because when we think about that idea of rescuing plants from pythium, sometimes there's a thought of, oh, well, once these plants are shaded, we're never going to rescue them. But this data set kind of showed that, maybe there is a small window where there's something we can target and get these plants back on track to try to rescue some of that yield.
Absolutely.
So, kind of wrapping up today was just a brief snapshot, but wanted to really just drive home that there's a lot of application where we can try to use some of these robotics, some of these new tools. I think the big takeaway here is there's just all this room for growth. And, we really are, I think just across everything in science right now, there's a lot of automation that's happening. There's a lot of changes in technology that are happening very rapidly that are opening new doors. So whether that's in disease prediction, in some of the way we approach our agronomic practices, I think we're going to continue to see more and more opportunities for automation to be able to try to use some qualitative replace some of our older qualitative assessments with new quantitative tools. And so I think it's a very exciting time for research and, appreciate Alison and Erin joining us today. And really, if you have any final thoughts to share with the group, but I think there's a lot of promise into the future of robotics in corn.